Uh, Good morning, ladies and gentlemen. It's 8 o'clock, and we start our now bi-weekly sessions. It used to be weekly. It's bi-weekly now. The wind beneath our wings, Bernie up, has retired, and so she's guiding us with her wisdom. And right now, our president is having a honeymoon. He's having honeymoon abroad, and I'm in going on a working thing with farmers. But we want to welcome you today, and we have a very interesting speaker. But before we go into that, let's have the opening ceremonies. Would be, of course, the prayer and the national anthem. And Vivian is here. She's now the wind beneath our wings. Vivian, can you start the process, please?
Yes, good morning, guys. So we'd like to welcome everybody. Today is very important to especially people who is in the planting business. So we'd like to, of course, firstly make mention about our corporate members, Chemical Corporation, Bounty Fresh, and so on. I'd like to also welcome the individual members, okay, which you can see in our screen. Okay, so welcome, guys. No, and then of course we'd like to make mention about our special partners institution, so you can see it in the screen. All right, and also we'd like to call our president to say something. Yes.
Yes, our president is on a honeymoon. He won't say anything. I'm the chair. I want to thank Robert Chupanco. He's probably the best entrepreneur in our group. He's the head of our ABCDF leads and needs. I have the pleasure to introduce our speaker. And I'm coming in from Iloilo meeting on farmer events. So Robert will take over after I introduce the speaker. So let me say who the speaker is. The speaker, ladies and gentlemen, forgive me for my voice. It's not my bedroom voice. It's just a hoarse throat. The speaker is Dominic Galimba. Dominic Galimba. His topic, as you know, is the business opportunity in seedling production. But I like to say that seedlings are the bottleneck in our agriculture development. So in addition to looking at this as a business opportunity, you must know that this is the key to our development. We don't have enough good seedlings. Now, let me tell you a bit about him. He studied, of course, in this area and went on to develop a very interesting career. Basically, he has something called a tissue culture, which is his very own, a Filipino tissue culture. It's his own. And I think at such a young age, he's pioneering already on that. He's consulting with so many different laboratories. And actually, this Filipino technology, Philippine culture is being spread slowly. He learned it initially in Rizal Technology University and grew the seed of knowledge into something that is really marketable now. So we'd like Dominic to start, instead of his lecture immediately, we'd like to say a couple of things. The first thing, of course, is our departed chair, backed by the wind beneath his wings, Bernie Yap, who has just retired, has said to us, this forum is not just for knowledge sharing. He wants us to get the knowledge from here and use it to spread to others so it can grow and do what the foundation wants, which is countryside development, creating jobs, and poverty alleviation. And we do that because our group here is not a group of students. We're professionals, thanks to the new board. Half of them are below 45, the other half are above. Before that, they were kind of old. Now with the young ones here, as a matter of fact, the board is 80% below 45 years old. Now with this energy, we connect you to banks, to government agencies. Sometimes we can do that because we're connected because of our experience and our connections. We do it faster. One example is someone took 5 months and we moved in and it took only 1 week. We connect you to networking, etc. So much for that. We'd like to say that's our mission. Dominic Galimba, however, will start because we work with him in between forums. We work with all our speakers because we need to spread their wisdom. So Dominic will start with 5 or 10 minutes about his personal life, how he got interested in this, and why he's passionate about it. After the 5 and 10 minutes, Dominic will give his so-called lecture. After
So
that,
without much ado, would like to call Dominic, okay, to carry on now. Thank you. Oops.
anyway,
Ernie,
okay. So anyway, with that, Robert, with your permission, I will give the floor to Dominic. I'll give the floor now to Dominic Galimba. Thank you, Robert.
okay, thank you, Ernie, also. Okay, carry on, Dominic.
Good morning, everyone. Good morning, and thank you so much, Agribusiness and Countryside Development, in partnership with Management Association of the Philippines Foundation. And I am Dominic Galimba po, and a tissue culturist since 2009 through the help of Rizal Technological University. Actually, it's my alma mater, no? And I started an academic laboratory setup together with my mentor. And after that, since 2013, habang nagkakaroon kami ng improvement on how we experiment everything in plant tissue culture, we conduct very simple setup in terms of practicality and simplified through alternative chemicals that we are using. We're not gonna use laboratory reagent or analytical reagent, which is too much expensive, no? We're just using agricultural grade chemicals, which is our fertilizer. Like 60 pesos per kilo lang, yun lang yung ginagamit namin, and very small amount lang yung ginagamit natin doon. And at the same time, even the equipments that we are using, and for laminar flow hood, it's actually very expensive equipments and other laboratory equipments na hindi natin masasabi na capable yung mga even students or farmers to put up their own laboratory. So with this kind of technology, we can actually teach even farmers can do their own laboratory. Yun yung pinakagusto namin, because for us, there's no such thing as high value crops in terms of agriculture if it's not came from tissue culture. Kasi through tissue culture, you do disease-free analysis. You do virus-free analysis or virus-free produce. So through tissue culture, they produce their own crops, their specific crops na masasabi nating high-end or high-yielding siya. So in this kind of technology, mas magandang may spread natin ito because of it's actually promising. Right now, in one of our laboratory in Pasig, which is in Harbest, we already have 300 different varieties of plants, from high-value crops and from ornamental plants, like for landscaping plants, no? Kasi ang concept is, lagi na lang bang paulit-ulit yung nila-landscape nating mga halaman? Bakit hindi na lang yung mga nanggagaling din sa Thailand na mamahali, tapos pamumurahin natin dito through tissue culture? And then, through conservation also, through conservation, we can do para at least magkaroon tayo ng conservation, especially sa mga endangered species natin. Not only categorizing what's endangered species, what's endemic, what is critically endangered. So at least nagkakaroon tayo ng sa habang kinakategorize natin yung mga species natin, at the same time, on the other hand, we are already conserving it, no? So yun po yung kinaganda ng alternative plant tissue culture. It is simplified, and in layman's term lang. The reason why we promote this kind of technology, kasi kahit anong equipments or laboratory na ginagamit mo, ang pinaka bottom line kasi niyan, yung halaman sa loob ng bote nag-grow ba. So yun yung naging concept namin. Kaya sa kahit anong paraan, even kitchen equipments, you can do your own laboratory, basta as long as tama yung ginagawa natin through our guidance po, using plant tissue culture alternative po. Yun po.
Okay, fantastic. Yeah, good. So now, shall we, do you have anything to, of course, to explain more in details about your tissue culture?
Yes, sir. Opo. Thank you,
Okay.
sir.
Yeah,
I think
Dominic.
I have my presentation now.
Yes.
Through Ma'am Vivian.
Yes, you can do your presentation, and then after the presentation, we will we'll have questions and answers. That's the formal meeting, no?
Sure.
And then after that, we'll ask, we'll give you, we'll close the, what do you call this, the formal presentation, and then we'll go to the informal presentation. That's from 9 o'clock to 10 o'clock. Up to
Okay.
what time? I hope you will have time for a lot of informal questions and answers that will be asked by our members and our guests. So, okay, go ahead, Dom. Yep.
Sure, Sir Robert. Thank you po. By the way, sir, is it okay if I'm going to speak in a bit in Tagalog and in
Yeah.
English para at least
Oh,
no?
yes.
Ma-deliver ko ng mas maayos yung aking
Correct. Correct.
information.
Sure. All right. Go
Thank you
ahead.
so much, sir.
Yes.
Okay po. Our topic for this morning is all about simplified and practical plant tissue culture. Why simplified and why practical? Yan yung i-discuss natin. Okay, next slide po. So in this topic, before we go to the business side of production of plant tissue culture, syempre, maraming hindi nakakaalam, especially what plant tissue culture is. Ano ba yung process ng micropropagation and its advantages and disadvantages. So at least before we move to the business side, ito munang mga simple information kailangan nating ma-attain. So yun po. Next slide please. Aseptic culture, maraming nalilito dito, no? Kasi when you say plant aseptic culture or plant tissue culture, they thought it's tissue culture also. But tissue culture is very broad. You can say it's animal tissue culture, human cell tissue culture. So napaka-broad niya. It should be plant tissue culture kung nagka-focus tayo sa propagation ng plant. And in vitro culture, meaning in vitro scientifically term as inside the container. So, yun naman yung mga nilalagay natin sa loob ng bote, which is, isa doon is yung the plant mismo or yung mga high value crops na nilalagay natin for us to clone. And what you call this? Micropropagation, because micropropagation from the word micro, maliliit na growing points of the plants, we are using apical meristem, we are using any parts of the plant, any growing points of the plant na medyo mas maliliit para mas mabilis siyang masanitize, no? Meron pa dyan, may mga microscopic like microscopic seeds, buto ng linga, mas maliit pa. Apical meristem, yung pinaka primordium ng halaman. So madidiscuss ko ulit yan mamaya. So kasi the reason why we're doing that kasi andun yung virus eh. Kaya kailangan ma-isolate natin yung virus to make our produce a virus-free plant habang kino-clone natin siya dun sa loob ng bote po. So aseptic technique, it is designed to provide a barrier between microorganism from the environment. Ito yung mga nasa paligid natin, mga nalalanghap natin, mga aerobic microorganisms or microbial spores, and the sterile cell culture depends upon the set of procedure to reduce the probability of contamination from these sources. Meaning to say, probability, because halos lahat ng ginagawa natin as tissue culturist, by chances yan eh. Kahit sabihin natin 1:5, sa 5:1, sa sa limang planting material na tinanim ninyo, isa lang ang tumubo, it's okay for us. Kahit magagaling na tissue culturist yung ginagawa, and then they will improve that na lang along the way. Kasi yung isang nag-grow na halaman sa loob ng bote, kaya natin yan paramihin, kaya natin yan gawing thousands eventually through tissue culture. Later on, i-discuss din natin yan. Aseptic culture, for me, it is not just a technique, but a skill of an individual for you to conserve your endangered species or your plant na gusto mo, for you to clone your plants, or culture plants aseptically. Basta malinis siya na nasa loob ng bote, na kahit septic yung area ninyo, siguro kami lang sa laboratory sa Harbest yung may tissue culture laboratory na anytime pwede kang pumasok sa lab nang hindi ka naka-lab gown, pwede kang mag-face mask, hindi ka kailangan kailangan yun, no? Pero dito kasi sa technology natin, ang pinakamahalaga, protected, may protection yung halaman natin sa loob ng bote. Sa kahit septic yung area mo, basta nag-grow yung halaman sa loob ng bote, dumadami yung halaman sa loob ng bote, it serves the purpose of our laboratory. So yun po yung skills kasi. Next slide please. Okay. So, isa pa po. Yes, thank you. Micropropagation process. So ito yung process natin, para at least magkaroon tayo ng idea kung paano ba dumadami yung tissue culture. Kasi minsan, sometimes some people told us, one plant into 1,000? How come? 1:1 lang yan kapag nilagay mo sa loob na ay sa paso. Pero why tissue culture you can produce 1,000 in one plant? So we have that process. So right now, number one, our number one, the selection of explant. Explant is our specimen, no? Yung planting material natin, yung mga growing points ng plants na gusto natin ilagay sa loob ng bote para paramihin natin. So, we're going to select our explants. May growing point ba yan? Nasang part ba yan ng plant? Nasa malupa ba yan? Visible ba yan sa naked eye natin? Kasi kung hindi naman visible yan, bakit pa natin i-tissue culture yun, di ba? Yung mga growing points na yan. So, kailangan muna natin magkaroon ng selection. For woody plant naman, yung pinaka-shoot-tip yung ginagamit namin yung mga soft part lang. Hindi yung makahoy na kasi mataas na yung possible source ng contamination niyan at mahirapan tayong mag-sterilize. And then number 2, explant sterilization. Ang explant sterilization po is, or specimen sterilization, we're going to sanitize the outer portion of the plant. So, aalisin natin or i-isolate natin yung microorganisms attached dun sa katawan ng plant natin para 100% mabubuhay siya without any microorganism intact dun sa loob ng bote. No? So, ibig sabihin, paano yun? Medyo mahirap yun. Yes, exactly. Dito isa sa mga binabayaran ng mga tissue culturist kung paano tayo maglalagay ng halaman sa loob ng bote. Even the procedure, binabayaran din yan sa amin po. Pero dito, sa sterilization of explant, ito yung sinasabi natin na kahit 5 or 10 planting material ang ginamit mo pero ilan lang yung tumubo, iisa lang, kung wala pa yung procedure no, ng UPLB or any tissue culture processes, it's okay kasi kaya nating maparami yan eh. Then once na dumami yan sa loob ng bote, number 3, which is our stage 1, nagkaroon na yan ng establishment, ibig sabihin the plant has already been adapting its new environment, which is nasa loob ng bote, inside the container, with 100% relative humidity kasi wala yung hangin. Umaasa lang yan dun sa gelling agent sa loob ng bote, which is agar-agar, plus our agricultural grade chemicals na kinombine natin para maging laki sa layaw or maging spoil yung halaman natin sa loob ng bote. No? Ang kailangan na lang ng halaman will grow the plant inside the bottle na. So, for about 2 to 3 months, yung ibang plant yun, no? Pero usually for banana kasi 1 month lang. But mag-focus na lang tayo for 2 to 3 months. After 2 to 3 months, the plant will establish its growth inside. Makita natin na nag-germinate na siya. That's the time that we're going to transfer it into another bottle. So dito tayo magkakaroon na ng computation. No? Like for example, ako, nag-clone ako ng plants. Isa lang ang tumubo sa stage 1 ko, it's okay. Kasi once na ita-transfer ko siya sa stage 2, which is our shoot multiplication, that's that thing na dadami na siya. We are using specific chemicals lang din, yung mga agricultural grade chemicals, plus plant growth hormones. No? Gumagamit tayo ng mga cytokinin hormones or phytohormones para mag-induce or dumami yung mga babies niya or clones niya inside the bottle. So yung stage 2 na yan, hindi lang yan isang pirasong stage 2. May cycle yan. Ang cycle niyan is up to 7 cycles if it is high value crops like bananas, sweet potatoes, vanillas. No, basta lahat ng mga high value crops, hanggang 7 cycle mo siya na pwede mong i-clone. Why? Kasi since we are using plant growth hormone, which is cytokinin or cloning hormone, dumadami yung halaman sa loob ng bote, and since it's phytotoxic, pwedeng magkaroon ng mutation yung halaman sa loob ng bote. Kaya hindi pwedeng yung high value mo magkaroon ng mutation. Kung ine-export mo yan tulad ng mga karatig bansa, tulad ng mga taga Mindanao natin na mga nagtitissue culture ng banana for export like Cavendish and Lakatan, they have this cycle para 100% yung quality ng plant nila is still the same. Kasi yun naman yung gusto nila for high value. No? So, like for example, ang one plant na nag-germinate sa isang bote, once na tin-transfer mo na siya for shoot multiplication medium, you will have at least, ang based on our experience, if it's banana, super dami na pag banana, other high value crops, hindi lang yan 1:1 kapag nilagay mo na siya sa cycle 1, which is the shoot multiplication stage natin. Usually, nasa minimum of 5 clones na yan. In just one bottle pa lang yan. So yung cycle 1 natin, after 2 to 3 months or usually 2 months, ita-transfer na natin siya sa panibagong cloning hormone ulit, cloning medium ulit. So yung 5, pwedeng maging 25 siya dun sa stages natin. So padami siya ng padami. So what more pa kung nandun na tayo sa cycle 7. Okay? So ganun yung process ng micropropagation. We can clone plants. We can produce a plant into 1,000 or more than 1,000 plants. It's either high value yan or ornamental. Pero kapag ornamental plant yan, tulad ng ginagawa namin sa laboratory, we have no unlimited cycles. No? Kung sakali, kasi mas magandang, mas matagal siyang naililipat. Why? Because ibig sabihin, mas nagkakaroon ng chance ng mutation, which is mas prefer natin kapag may possible new mutation. Ibig sabihin, new variety of plant yan. So kung ornamental plants yan, may bago tayong variety ng halaman na na-produce na that we can register it, no? So yun po yung advantage ng micropropagation process while cloning pa lang. Then after that, stage 2, we're going to transfer, like for example, we already have this kind of quantity na gusto na natin siyang ilabas sa loob ng bote, ilalagay na muna natin siya sa panibagong culture media na instead na merong shoot multiplication hormone or cytokinin hormone, we're gonna use rooting hormone naman. Usually, familiar naman kayo siguro sa mga Anaplas, Hormix, Miracle Grow. Yun lang yung dinadagdag po namin para magkaroon ng ugat yung halaman inside the bottle. Kasi hindi tayo pwedeng maglabas ng halaman kahit gaano pa kadami yan pero wala yung ugat, hindi yan mabubuhay sa labas. So through rooting stage, diyan na magkakaroon ng establishment ng ugat or magpo-produce ng true roots. Then after true roots production, we're going to transfer it in acclimatization stage. Kumbaga sasanayin na natin yung halaman sa natural environment niya, which is eto na, from 100% relative humidity inside the bottle, we're going to transfer it in ang ginagawa namin, seedling tray lang po, tapos kinukuluban namin, and then for how many days, dahan-dahan namin tinatanggal yung cover para nasasanay na siya, hindi siya nabibigla, nasasanay na siya sa panibagong environment niya, which is 60% relative humidity. Then after that, inaalagaan na lang natin siya. We're also using same chemicals as foliar fertilizer. Yun din po yung ginagamit natin para hindi na tayo bibili ng chemicals natin. Para once na nag-grow na siya for desired plant, around 3 months, we can sell our plants. So in just 1 year, you can produce your own plants. From one plant, you can already produce thousands. No? Dumidepende na lang sa tissue culturist kung gaano kadami yung capability niya. That's why sa mga staff namin, ang nire-require namin every day, we require them 80 to 10 flask per day lang per head. Yung 80 na yan, hindi yan 1:1 pa rin sa loob ng bote. Kino-clone pa rin namin yan. Kumbaga, nasa 8 to 10 plants per bottle kami. So usually ganun yung output namin. Yes, meron kaming contamination. Kahit anong laboratory may contamination. No? Pero nami-minimize natin yan kasi meron tayong protectant mismo dun sa loob ng bottle na makikita nyo later on. May picture po ako later. Then after that, that's the time na pwede na nating ibenta yung plants. Same as 100% true to type siya na kung saan natin siya kinuha. Which is yung mga sanga lang ng plant or growing point ng plant. Yun lang po. Pero may instances din po na kapag seeds ang gagamitin mo, like for example, papaya seeds, no, meron yan, 3 klase yan, lalaki, babae, tsaka hermaphrodite. We never know kung anong klaseng lalabas yan if it's seeds. Pero if we want to produce hermaphrodite papaya, 100% through cloning, kuha tayo sa growing point ng hermaphrodite papaya and then we do cloning. So lahat ng produce mo, 100% hermaphrodite papaya na through tissue culture po. So yun yung advantage ng plant tissue culture kung gagamit tayo ng planting material through the part of the plant. Next slide po. So dito po may mga advantages and may mga advantages tayo ng tissue culture, no. Number 1, you can produce numerous propagules. As I've said earlier, masyadong marami. Minsan kami na lang din ang sumusuko kasi hindi namin alam kung ano yung uunahin namin ipaprioritize sa laboratory. Dumidepende na lang po sa kung ano yung request ng mga clients namin na paparamihin namin. Pangalawa, you don't need any too much space for the laboratory, no? Basta as long as kung nasa loob kayo ng bahay, actually sa condo nung hindi pa nanganganak si Mrs., nagput up ako ng laboratory. And dito lang sa condo, ay masasabi kong efficient naman siya kasi nakakapag-produce ako and then tina-transfer ko kay Harbest para doon ko na lang siya kinukulture or ini-incubate. So if space efficiency kasi kailangan mo lang dyan ng shelf, kahit mga 5-layer shelf. No? Hindi mo kailangan ng malaking nursery or nursery bench for your tissue culture laboratory for culture room. And other than that, meron din tayong mga rooms mamaya na ipapakita ko po. Propagation can be done all year round. Ibig sabihin, hindi naman to seasonal eh. So, pwede natin i-propagate yung mga plants na gusto natin kahit sa loob ng bote. Seasonal yan, ngayon lang yan tutubo. No. Kapag tissue culture, iba yung buhay niya inside the bottle. So, kumbaga, iba yung kwento if we're talking about tissue culture. Next is disease-free plant produce. Lahat ng plant, basta tissue culture yan, it's actually disease-free yan. Makikita natin on through our naked eye na contaminated yung halaman natin kapag may mga amag yung loob ng bote natin. So, dun pa lang, possible na magkakaroon na rin siya ng disease, no? Pero, as long as walang contamination yung halaman at nag-grow siya, it's actually disease-free plant produce and even virus-free. Kasi we are isolating the virus dun sa vascular vessel ng plant. Kaya masasabi nating virus-free din yung mga halaman natin. Maganda ang output natin, especially for the plant, kung virus-free siya. Mabilis ang produce mo, maganda ang output mo, maganda ang produce mo, pati yung mismo mga fruits mo. Conservation and free from environmental risk. Dito, kaya as I've said earlier, in terms of conservation or conserving plants, you're not going to get the whole plant in the wild, no? Kasi most likely mga poachers or illegal loggers, hindi nila alam may mga endangered species pa lang nakakabit na halaman dun sa puno, di namatay na din habang kinukuha nila. Hindi naman nila wala naman din silang pakailan dun kasi ang gusto nila makuha lang yung logs or yung mga kahoy. Pero kawawa naman yung mga endangered species natin. So paano natin mapapalagaan yun? Number one, we cannot get the whole plant, no? Take it out from the wild and then you conserve it. Paano yun? So sa amin, ang ginagawa namin, kukuha lang kami ng part of the plant. Kung may seeds, why not? And then, ilalagay lang namin sa ziplock na may tissue paper plus distilled water, transfer sa laboratory, and then conserve, clone. Then, once na meron na kaming desired quantity, balik natin sa wild. So at least, andun yung mother plant na pinagkunan namin ng planting material or ng specimen, at least, napaparami pa natin siya. So, hindi siya totally free risk sa environment natin. Transportation-wise and not subject to plant quarantine office. Why? Yung mga may lupa. Sa atin, through tissue culture kasi, mga bote yan na merong artificial soil or medium na yun yung pinanggagalingan ng pagkain niya sa loob ng bote. So, hindi siya totally contaminated or wala siyang masyadong disease or wala siyang peste kasi walang contamination yung plant na nasa loob ng bote. Kaya hindi siya kailangang i-quarantine pa. So very safe siya and at the same time super clean niya kasi nasa loob siya ng bote. No need for daily watering and spraying of pesticides while inside the flask or bottle. Ang kailangan lang natin is number 1, light. It should be 12 hours to 16 hours na nasisinagan siya ng liwanag, natural light or artificial light. And then meron lang tayong specific temperature na mini-maintain, right? Like around 26 to 28 or 27 degrees Celsius. Kung pumalo na yung kwarto namin sa laboratory ng Harbest ng 28 to 29 degrees Celsius, that's the time na binubuksan lang namin yung aircon. So meaning to say, since we're not government na 24/7 pwede namin buksan yung aircon kasi may fund naman din sa kuryente namin. Sa amin kasi hindi ganon kasi dadagdag din pa kami sa costing pagdating sa mga plants na ginagamit namin. So meaning to say, hindi kailangan yung halaman laging nag-aircon. Mas maganda siguro yung nagtatrabaho na lang in-aircon natin. So mas gaganahan pa siya at mas less pa yung aerobic microorganism sa katawan niya. So yun yung advantage ng tissue culture, no? And then syempre, lastly, less care or attention between subculture. Subculture, ito yung nililipat mo yung halaman sa panibagong bote, no? So ibig sabihin, hindi mo kailangang araw-arawin yung pag-check diyan. After 5 days, check mo i-check, okay lang. Kung umalis ka galing sa abroad, tapos pagbalik mo, double check mo kung may contamination, kung nabuhay, kung namatay. As long as controlled yung humidity natin, controlled yung light natin, yun naman yung concern natin, no? Even sa loob ng bahay mo may plant ka, kailangan din yan ng what you call this, ng ilaw or ng sunlight para mag-photosynthesize siya. Ganon din yung halaman sa loob ng bote. Less care lang. So, no worries about that. Next slide, please. If we have benefits and advantages or pros, we have also disadvantages and cons sa tissue culture. Number one, you are required to train in aseptic culture, no? Kailangan may background ka at least ng plant tissue culture kasi sasabihin yung iba, based on our experience, why are you buying tissue culture plants? It's very sensitive and it's really hard to grow outside. Pero hindi nila alam yung part ng pag-acclimatize kasi ng halaman. Kasi nasa loob siya ng bote, ilalabas mo, ilalagay mo kagad sa paso. Patay talaga yung halaman yan. Why? Because lahat ng halaman or all the plants inside the bottle, the plants, open lahat ng stomata niyan sa ilalim ng dahon. No? So once na ilalabas mo siya agad na hindi ka gagawa ng acclimatization process, so ang tendency sa plant, kahit 10 minutes lang yan, patay yan. No? Malalantayan. Para lang din tayo, like for example, nakanganga tayo ng 10 minuto, matutuyuan din tayo ng laway eh. So eventually eh. So ganun din yung plants, no? Kaya mayroon tayong specific process on how we acclimatize the plants. Kaya hindi natin masasabi na sa una, yes, kailangan. Kasi syempre, kailangan din nating alagaan paglalabas ng babies natin galing dun sa sinapupunan, papunta mismo dun sa soil, di ba? Kailangan may alaga pa din siya. So, yun po. And then, space requirements. Yes, kahit maliit lang na kwarto, okay lang. Pero, syempre, kailangan pa rin natin ng required space for that. Because through tissue culture, kahit na nasa loob yan ng bote, may mga space tayong i-require dyan. Mamaya, i-discuss ko din sa makikita niyo po. Labor intensive due to laboratory maintenance, yung mga chemicals na ginagamit natin, laboratory staff na pinapasahod natin. That's why we require them to produce at least 80 to 100 flask per day. 3 times a week yan nilang ginagawa kasi magluluto pa sila ng mga culture media din and inventory din sila, general cleaning din sila. Kaya 3 times a week lang namin sila pinapaproduce ng clones namin. Para doon namin makukuha yung pansasahod namin sa client, sa mga staff. Next is the contamination occurrence. Sa kahit anong laboratory na kinoconsult namin, even Thailand and Malaysia, lagi nilang sinasabi, sir, kasi we have 5 to 10% contamination, especially in rainy season. So dito sa atin, we invented a copper fungicide na hinahalo sa pintura para hindi papasok yung mismong moisture at even fungal spores na nanggaling sa labas sa mismong laboratory mo. Kaya nagkakaroon tayo ng mga mushroom sa bahay, sa mga dingding natin, di ba? Kapag habang tumatagal, nagkakaroon ng moisture. So, ang tendency, galing yun sa labas na nauulanan, pumapasok yun dahil sa lamig. Kaya nagkakaroon ng mushroom yung gilid ng mga bahay natin. So, on top of that, kailangan lang natin ng Nordox. I'm not sure if you're familiar with Nordox or copper fungicide. Mura lang yon. Nasa 25 pesos per 20 grams lang yata yun sa Shopee. Ihalo mo lang yun sa 1 gallon of 1 gallon of paint. So, meron ka ng copper fungicide paint na pwede mong ipampintura hindi lang sa laboratory, no? Para maging long term yung bahay mo, yung area na gusto mong pagputapan ng lab na wala masyadong fungi or fungus na pwedeng tumubo dun sa area na yun. And lastly is, yes, long-term investment. Kasi tulad ng sabi ko po kanina, long-term investment kasi you just have to wait from one plant. Hindi lang yan one plant, ilalagay mo sa loob ng bote. After germination, nag-clone ka lang ng 5, ilalabas mo na. Hindi, sayang naman yung in-invest mo, no? Na chemicals, na laboratory. Kung ganun lang din yung process. Paramihin na muna natin. It's actually long-term, no? Depende sa specific crops na ifo-focus natin. Like for example, sa banana, super bilis niyan. Even 1 month pa lang, nililipat na natin siya. So after a year, you can already produce thousands of it. Depende sa dami din ng staff ninyo. Kasi doon tayo mamaya magkakaroon din tayo ng computation po kung paano natin ma-attain yung specific quantity for that particular quantity of plants. Yan po. And next. Next slide, please. Yes. So dito po sa Harbest tissue culture natin sa Pasig and even sa Halahala, we have our media preparation room. Next slide po. Sa media preparation room namin, we have this kind of area lang. Meron lang kaming bintana and then makikita nyo, meron lang yung lutuan namin, we are using bigger pressure cooker para at least meron tayong by quantity or commercial agad yung magiging produce ng output natin sa nagagamitin natin for cloning. So as you can see, very simple lang siya. Hindi siya, pati yung mga trainees namin, yung mga visitors namin, hindi kailangan naka-lab gown. As long as alam natin kung ano yung mga kailangan lang ilagay sa loob ng bote, mga chemicals na ginagawa natin. And even our storage, basta as long as hindi siya hindi siya direct from sunlight, it's okay. No worries. Basta hindi mo kailangan ng ref for your chemicals, no? Palagay ko naman yung mga abono ng mga farmers natin, hindi naman nila nilalagay sa ref, diba? Pero they are still using it from time to time. That's why ganun din tayo. Basta hindi lang siya direct sunlight, especially sa mga hygroscopic fertilizers like calcium nitrate, masyado siyang mabilis yan matunaw or mamasa. So ang tendency niyan, pwedeng mag-liquid siya kapag masyado yang mainit. So ilagay mo lang sa hindi naaarawan or naiinitan, kahit sa mga shelves lang, it's okay, no problem. So yan po yung media preparation area natin. Next. Next slide, please. And then our transfer room. So sa transfer room na to, some they call it dissection room and inoculation room. So ito po yung aming transfer room, no. We are using transfer cases. As you can see, yung mga gumagawa, hindi rin sila naka-lab gown. Even yung iba hindi rin naka-mask. Kasi ang kailangan lang natin, meron lang tayong meron lang tayong specific process kung paano ka gagawa muna dun sa workstation mo. Para aseptic pa rin yung ginagawa natin. As you can see, makikibalik na lang po sa isa. Previous slide, please, ma'am. Yes. As you can see, ito pong nasa gitna ng picture, that's our blower box. That blower box is actually, yes, thank you po, the heart of our transfer case. So yung bawat transfer case na yan, pwede natin siyang ihalin tulad sa laminar flow hood. Because number one, laminar flow hood is very expensive, super expensive niyan. Pwede ka naman din gumawa. Pero itong blower box, yan yung puso niya kasi andyan yung HEPA filter, andyan yung UV, andyan yung may pre-filter pa tayo, pati turbo type blower na it can accommodate up to 5 transfer cases. So meaning to say, sa isang blower box na yan, kaya niyang kaya niyang magbuga ng hangin, ng clean air sa 5 transfer cases. So para ka na din bumili ng 5 laminar flow hood na around 300,000 ang isang laminar flow hood. So ang laki ng natipid natin. So sa gantong technology. So ganyan din ang ginagamit namin kaya nakapag-produce kami ng 300 different varieties of plants with around 26,000 bottles of different varieties of plants na kaya nag-extend na po si Sir Toto Barcelona ng kanyang laboratory sa Halahala, Rizal po. Next slide, please. So that's our trainees, no? we train people na sa mga even farmers. Kaya yung iba nagta-train sila, hindi naman wala. I don't have any idea about plant tissue culture. Is it okay if I'm going to join your training? Yes, no problem. Kasi halos lahat naman ng mga terminologies na ginagamit natin dito is layman's term lang. So they can understand. So we have Tarlac Agricultural University students and Nueva Vizcaya state universities, other SUCs and LGUs nag-train na rin sa atin, and even farmers and students na gusto ko kasi magkaroon ako ng study or research with this kind of alternative lab. So nakakatulong din tayo makapag-promote ng gantong technology sa kung ano pa yung naiisip nilang gustong gawin through tissue culture, no? Kaya ang laking bagay ng training na to para at least mamulat tayo sa gantong teknolohiya, especially yung mga academe approach, no? Yung mga alam nila high-end yung laboratory, it's not necessarily kailangan. So kahit gantong laboratory lang, it's okay, it's productive. Ang magiging result natin makikita pa din which is yung mga nasa loob ng bote na halaman. Next slide please. Yes, this is our culture room naman po. Our culture room, dyan naman po natin inilalagay or we are storing all the cultures or plants in that kind of shelves. So, dun po sa culture room natin, yan po yung incubation area or incubation room naman na tinatawag. So, yan po, si Harbest din ang nagpa-fabricate din ng mga culture shelves na ginagamit natin or na pinag-iistakan natin ng mga bottles. So, next slide po. Yes. So, as you can see, may mga microwavable box din tayo na nandyan sa culture shelf. Why? It's actually ina-acclimatize na natin siya, sinasanay na natin siya sa environment. So, kahit na, sabi ko, instead na ilabas mo yan sa grow out area, might as well ilagay mo na lang din yan sa culture room. Hindi naman yan totally mako-contaminate kasi protected yung ating mga cultures. So, at least at the same time, mas mag-a-adapt siya dun sa environment kasi hindi siya mabibigla dun sa init. So, sa gantong setup, you can produce your own laboratory. Actually, we started 40 bottles lang way back 2019. 40 bottles lang. And right now, since 2024, we already have 23,000 bottles continuous. And then at the same time, may mga nabebenta rin tayo. May mga nagre-request na rin ng mga specific crops na gusto naming i-produce para sa kanila. May mga ornamental plants din tayo na pinuproduce on our own. Yung mga rare plants na galing sa Thailand. And at the same time, we conduct research and development through DOST or Department of Science and Technology, Philippine Nuclear Research Institute, for us to produce new variety of plants. No? Kasi sa Thailand, they are using chemical mutation or chemical mutagenesis. Kaya when you say variegated plants na kakaiba at mahal, nag-start yan Thailand because they are doing that long time ago pa. Pero chemically induced, meaning to say, when you say chemically induced mutation, it's actually point mutation, meaning to say, point lang siya. Isang part lang yung nag-mutate, hindi the whole plant. So meaning to say, hindi DNA ng halaman yung nagbago. Mismong itsura lang. Kaya minsan, yung mga sellers like 4 years ago, yung mga umusbong ang mga plantito-plantita natin, bakit nagkakaroon ng reversion yung mga halaman na binili kong mahal? From variegated, nagiging normal. Yes, it's because of point mutation. There's a tendency na because of that, no? Pero hindi rin natin masabi, maybe it's because of the fertilizer that they are using, which is masyadong mataas ang nitrogen, kaya nagtatago yung variegation. Possible yon. Pero through physical mutation or mutation breeding through gamma irradiation, anything is possible. Anything that you can imagine, everything that you can imagine na pwedeng mangyari sa plants, pwede kang makapag-produce ng mataas na content ng sugarcane, mataas ang mawala yung kate or yung calcium oxalate of gabi, no? Or pumunggok yung halaman. It's not just a variegated plants. Through gamma irradiation kasi, makakapag-produce tayo ng sarili nating plants. So ganun din yung ginagawa natin. And at the same time, mas mapapababa yung waiting time natin. Kasi when you say gamma irradiation, you have to wait 4 to 6 years, no? For you to have your possible mutated plants. Pero through tissue culture, you can wait up to 1 to 2 years lang. Meron ka ng mga different cultures na mutated. Kasi kaya nating i-clone yan eh. Kaya nating paramihin yan eh. Even cells kaya mag-mitotic division yan sa loob ng bote. Yun yung isa-subject natin for gamma irradiation. So, para lahat yun, once na nag-survive dun sa particular gamma, there's a big tendency na magkakaroon na tayo ng bagong variety ng plants. At the same time, it's not only point mutation. Since DNA na nag-recombine yan or nag-repair na DNA yan, ibig sabihin, nabuhay yan pero hindi na yan bumalik sa dati yung DNA helix nila. So, ganun po yung isa sa mga research and development na ginagawa natin para at least after 2 years, meron na tayong bagong variety ng plants na pwedeng ilabas. Na masasabi